Hello students, welcome to Capman Academy. Today we are going to discuss about food nutrition topic, which is the carbohydrate. So carbohydrate, it is one of the essential major biomolecule, right? So as we know, there are major biomolecules which are present and very important for the growth of the organisms, right? So these major biomolecules, one is carbohydrate, one is your lipids, one is protein, and one is nucleic acids, right? So they all are very important for the animal's growth. So now we are going to discuss one by one how these major biomolecules are actually synthesized, what are their structure, and how many types are there for them, right? So first we are going to discuss about carbohydrate. So as we know, carbohydrate, as we speak in the plants, they actually synthesized by a process which is called photosynthesis, which is a chemical reaction in which the carbon dioxide and water react with each other in the presence of sunlight, and it produces glucose, which are your sugar molecules, and also liberates the oxygen, right? So glucose, which is being synthesized here, it has an empirical formula which is called CnH2nON. This one is very important, right? So as the glucose sugar molecules are being synthesized in the plants, when organisms consume these plants as diet, so these sugar molecules are actually being used for the organisms, as we speak, for the human being growth, right? And they are being used in different type of biological processes, right? So first of all, these carbohydrate, in what type of the plants they are present and what type of functions they have, why we actually needs the carbohydrate in our diet. So first of all, carbohydrates are basically present in high amounts in your different type of grains, different type of food, right? As you can say, the bread and your potatoes, they all are rich in the carbohydrates. And the benefits of the carbohydrates in the diet is they actually have the most important energy molecules. They provide the energy for the body to work properly. It helps to sleep better, helps to prevent the diseases, useful to control the weight, helps to keep memory sharp, best nutrient for the athletes, improves the digestive system, prevents the blood clots, provide fibers to the body, and uplifts the mood. So it has really so many benefits for the human beings, right? So now let's discuss about what is the structure of carbohydrates and what are the types of the carbohydrates, right? So as we speak, first you should know about the carbohydrates. What are they actually are? So carbohydrates, they are defined as number one, hydrates of carbon, hydrates of carbon due to the presence of specific ratio of carbon and water, right? So on the basis of this, they are called hydrates of carbon. They are also defined due to the presence of the functional group they have, in which they are known as polyhydroxy, aldehydes, and polyhydroxy ketones, right? Or the compounds which upon hydrolysis produces these type of compounds. They are all called carbohydrate, right? So according to their structure, carbohydrates are actually being classified into different categories. The major categories are number one, the monosaccharide, which means only one sugar molecule will be present here, right? Next category is called disaccharide, in which two sugar molecules will join each other and they form disaccharide. Two sugar molecules will be present, right? The next one is called oligosaccharide, in which more than two sugar molecule will join to each other and they would be from 2 to 10 molecules. So this chain is called, small chain is called oligosaccharides. Polysaccharide is the lengthiest chain in which more than 10 sugar molecules will actually join together and they form the or produces the complex polysaccharides. Right? So according to their structure, they are being categorized, and the examples of them are, in the case of monosaccharides, glucose is the single sugar molecule, fructose and galactose, they all are the single monosaccharides. Right? If we talk about the disaccharides, then it is sucrose, lactose and maltose, which means two sugar molecules will be joined to each other. In case of oligosaccharides, they have more than 2 and less than 10 sugar molecules present in the chain, and those are called raffinose and your stachyose. In case of polysaccharides, you have starch, glycogen and cellulose as the example. Right? So if we say, in sometimes disaccharides are also actually being involved in the oligosaccharides. Right? So disaccharide here, you can say them, they are also the oligosaccharide in which two sugar molecules are actually being joined together. So now we will discuss one by one about these sugar molecules, how they are actually being synthesized and how these sugar structures are actually being formed. So as the name suggests, monosaccharide means only one sugar molecule will be present in this one, and this one is called simple sugar. Simple sugar, right? Which means this type of sugar, it cannot be hydrolyzed. There won't be any hydrolysis up to this point, right? And its empirical formula is CnH2nOn. This one is very important. Sometime question can be asked from here, right? And the example we had already discussed about them. So in case of monosaccharides actually they are categorized according to their number one, the carbon atom which are present in the structure, right? Second, the functional group which are present in your sugar molecule, and the third, the numbering system of the carbon atoms. Right? What it means? Carbon atom means your sugar molecule, how many number of carbons it has. If it is having the 3 carbon atoms like this, then this one is called triose. If it is having the 4 carbon atoms, then this one is called tetrose. So if it is having the 5 carbon chain, then this one is called pentose. Right? And if it is having the 6, then this one is called hexose, and so on. So on the basis of carbon atoms, your monosaccharides are actually being derived, right? Next comes the functional group. As we talked about, your carbohydrates, they are actually polyhydroxy of aldehydes. For aldehyde, this one is the aldehyde group, or the polyhydroxy of the ketone group. Right? So which one of the functional group will be present in the carbohydrate? According to that, your monosaccharides are being synthesized. So they are categorized into 2 categories. So the molecules which contain the aldehydes, those are called those are called aldoses, and which contain the ketone group, those are called ketose sugar. Right? Next one comes is the numbering of the system atom. As we speak, it is a chain of the carbon atoms. Now, how actually you will decide that which one of the carbon would be the one and which one carbon would be the last one? So that also need to be defined. So numbering system of carbon atom means that your carbon chain numbering will start from that point where your functional group will be present. So where the functional group will be present, that carbon would be your first carbon, then second, then third, then fourth, and then fifth, and so on. Right? So these are the some common rules which actually being followed during the monosaccharide synthesis, and they are the very important one. Number one is the carbon atom. How many number of the carbon atoms are present in your carbohydrate molecule? Next is the functional group. Which one functional group is present? And next is the numbering system of the carbon atoms. Right? So, for example, if we take an example of glycerolaldehyd, which has 3 carbon atoms, right? So this one is your triose. This is the triose monosaccharide, right? Now it has aldehyde group here in the first carbon, and the next there are hydroxyl and hydrogen group and CH2OH group. So this one is the structure of glyceraldehyde. Okay, now because it contains the aldehyde group, this one is your aldose sugar. Right? So this is how you can say the first carbon would be this one, second carbon would be this one, and third carbon would be this one. 1, 2, 3. This is the numbering system. Carbon atoms, how many carbon atoms? 1, 2, 3. Then this is called triose, right? And the functional group, because it is having the aldehyde group, then this is called the aldose. So this is the simplest structure for the glycerol aldehyde, and this is the basic simplest sugar molecule from which your carbohydrate or your monosaccharides are actually being derived. So if sometimes question can be asked that your monosaccharides are actually being derived from what? So they are your triose sugars. So if they are talking about the aldehydes, then it would be glycerol aldehyde. And if they are talking about the ketose sugar, that would be your dihydroxy acetone sugar. So they both are the simplest trioses or simplest monosaccharides. From them, their tetroses are being derived. Then pentoses are being derived by the condensation reactions. Condensation reactions. Right? So this is all about the monosaccharides. So now in carbohydrates, other than this features, they also have include some processes, right? They also have some important processes which are going on in the carbohydrates. Why? Because of their structure. So first of all, as we all know that carbohydrate, or if we simply say when a carbon atom, it is surrounded by different type of 1, 2, 4, 3, 4 molecules, then this type of carbon is called chiral carbon, right? So this chiral carbon, it is actually present in the carbohydrate. ठीक है? So due to the presence of this chiral carbon, your carbohydrate, they have the chirality in their structure, and due to this chirality, they actually form different type of isomers, and they follow stereochemistry. Stereochemistry क्या है? Let's discuss about it now. ठीक है? So first of all, I hope you understand about the chiral. Your one particular carbon atom will be surrounded by the different type of groups. Then in that case, your carbon would be the chiral, right? If it is having the same molecules 2, 2, 1 and 3, then this won't be your chiral, this would be your achiral molecule. And these type of structures, they are actually are your non superimposed structure, non superimposed structure, which means if you are trying to align them together, then there won't be the mirror images for them. How? Because when you actually put these chiral molecules in a solution, right? When you put these chiral molecules in a particular type of solution, that in the presence of light, they actually changes their plane. Okay? So in case of carbohydrate, this change of plane occurs at the hydroxyl group. Let's have an example here. So let's talk about the glycerol aldehyde. We have a CHU molecule, carbon 1, then hydroxyl group, and then one hydrogen, and then one CH2OH group. So as you can see, 1, 2, 3, and 4. All the components from this carbon are different. They are attached differently. So this one carbon, number one, is your chiral carbon, right? So when you actually put this type of molecules in the aqueous solutions in the presence of light and impose a light on these solutions, then this plane of the hydroxyl atom molecule will change, right? It's like that this hydroxyl carbon, if it is in the right side, then upon the incorporation of the light, this hydroxyl molecule will change its plane into the left side, right? So if it is in left side, it will change into the right side. And according to this change in the plane, because of this change in the plane, these sugar molecules are actually formed different type of configurations, and these configurations are called your isomers, right? So this configuration, it could be number one, your right configuration, which are called D configuration molecules, which means your hydroxyl molecule will actually change its plane into the right direction. Next configuration, if the hydroxyl molecule is changing its configuration into the left side, then these are called L configuration. And the change will be the left side. ठीक है? So due to the chirality, there is the stereochemistry present in the carbohydrates, and due to that chirality, your plane of hydroxyl group will change in the carbohydrates, and they will form two type of the molecules, D configuration molecules and L configuration molecules. So this is the reason, that is why you have two different type of the isomers in the carbohydrates, right? So now, in this case, we are talking about only the one carbon atom. Now, what will happen when there are multiple carbon atoms present in the carbohydrate molecule? As we know, your carbohydrate is a simple carbon atom chain, and in that chain, there is a possibility that the chiral carbon would be 1, it can be 2, or it can be 3. Okay, so there can be multiple chiral carbon can be present in a monosaccharide molecule. Now in this case, when there are multiple chiral carbons present in your structure, how you will decide the configuration of your molecule, right? So in this case, keep that in mind. The farthest one chiral carbon, or the chiral carbon which is near to your terminal chiral carbon in the sugar molecule, that will actually decide the configuration. So in this case, if you have 6 carbon molecule, right? And here you have hydroxyl group in this side and hydrogen atom this side, and this is your chiral carbon, right? So this chiral carbon configuration would be D configuration, because this one carbon would be the farthest of the chiral carbon, right? So this is how you will decide when there are multiple chiral carbons are present in the carbohydrates, right? I hope everything is clear until now. Now next, move on to the other type of processes which occurs in the carbohydrate. So this is the one of the process, that one is called stereochemistry. And in that case, different type of isomers are formed. Next one is the formation of apimers. Apimers, or the process is called apimerization. So this one actually a simple process. In this case, your whole structure of the sugar molecule will be same, and it will differ only one carbon position. All the skeleton of your carbon sugar will be same, but only it will change in the one carbon position. How? Because at that particular carbon position, your hydroxyl plane will be different, right? So when this type of phenomena occur in which all the skeleton of your sugar molecule is same, but they differ only one carbon position, then that type of the molecules are called apimers. right? And these apimers are basically glucose apimers. Glucose apimers. So for example, if we say galactose is apimer, galactose is apimer of glucose at carbon 4 position, which means 1, 2, 3, 4, fourth number of carbon plane of hydroxyl group will be different from the glucose molecule, right? Next example is the mannose. Mannose also is a monomer of glucose, but it is also different in the position. It differs in the C2 position. So sometime question can be asked according to their position, carbon position at which they actually behave like an apimers, right? Or it can be asked simply which one of them are the apimers of glucose. So keep that in mind. Galactose is the glucose apimers or mannose is the glucose apimer, but mannose is not a galactose apimer. ठीक है? This you need to keep that in mind because sometimes question can be asked from here. So this is about the epimerization. Next process which occurs in the carbohydrate that is the change into the form. So the chain structure of the carbohydrate, this one structure is called your Fischer projections. Fischer projections. Right? So when the chain will become very lengthy, in the solutions, it's actually react with each other and it form a cyclic structure. Right? So functional group carbon atom, it actually react with your fifth number of the carbon atom and they form a cyclic structure like this one. So these structures are called Haworth projections. Haworth projections. Okay? And these are the cyclic form. Now these cyclic form, if there are your 5, then these are called pyranose. 1, 2, 3, 4, 5. If there are 3, then they are called, if they are 5, they are called pyranose, and for the hexoses, they are being called as pyranoses, and for the ketoses, they are called as furan. Right? So now, when these Haworth projections actually forms, in this case also, there are different type of configurations occur. Okay? Configurations at what position? Configuration according to the carbon first hydroxyl group position. Right? In this case also, your hydroxyl group, it actually change its plane. Okay? And due to that, it will form or produce different type of anomeric structures. Right? So why these structures are called anomeric? Because they are actually being formed at the first carbon. So this structure is actually being decided by the configuration of the first carbon of the Haworth projections. Right? So in this case, if your Haworth projections, they have the hydroxyl molecule above the carbon plane, then this type of anomers are called beta anomer. Right? And if your hydroxyl molecule is actually lower the carbon plane, then these type of anomers are called alpha anomers. Right? This is your sugar molecules and this is your oxygen. Right? So these are called your beta anomer and alpha anomer. Sometimes simple question can be asked, what are the anomeric structures? And this one is carbon 1, where these anomeric structures are actually being formed. What is the reason for them? It is the carbon 1 where the hydroxide group plane is actually being changed. Changing how? in the aqueous solution, when you're keeping your sugar molecules in the aqueous solution and passing the light through it, right? So now I'm talking about the aqueous solution. So when you actually add your chain structures in a solution, first thing happens, they will start to make the cyclic form. And in this cyclic form, it will form two type of anomers, alpha anomer and your beta anomer, right? So during this reaction, during the interchange of chain reaction to the cyclic reactions, right, your alpha anomers and beta anomers starts to form until they reach at a equilibrium. Until that, there is a interchange of alpha and beta anomers. There could be higher concentration of the alpha anomer and higher concentration of the beta anomer. There could be the lower concentration of beta anomer, higher concentration of the alpha anomers, right? Until they reach upon a equilibrium state. So this process of interchange, interconversion of the alpha and beta anomers from the chain to cyclic form is called mutarotation. It's called mutarotation. So this is the another process which occurs in the carbohydrate. So we talked about three processes. Number one is the formation of isomers, D and L configuration isomers. Second process is the epimerization, and third process is the mutarotation. Mutarotation, right? So in case of mutarotation, I am repeating it again. So in this case, your chain formation of the sugar molecules, it actually in the aqueous solution presence making the alpha enomer and your beta enomers, right? And it will interconvert with each other until they reach an equilibrium point. So this interconversion of the anomers in our aqueous solution is called mutarotation process. Mutarotation process. Okay? So this one is also the important one process which is present in the carbohydrates. Right? So this is all about the monosaccharides. We had already talked about the simplest structure of the monosaccharides, right? How they are actually being synthesized by the process of condensation, and in that condensation simplest trio sugars are actually responsible for the synthesis of tetroses, your pentoses, and then your hexoses, and so on. Okay? After that, we talked about some important processes which present in the carbohydrate. So number one is the stereoisomer. Stereochemistry is also present in the carbohydrates due to the presence of chiral carbon. Then further we talked about epimerization, and then further we talked about the mutarotations. Right? Now, in some cases, your simplest monosaccharides, simple structure of the sugar molecules are actually altered, or you can say their structure is being changed at different locations, right? So due to this alteration, these type of molecules are called monosaccharide derivatives. So your monosaccharides are also have monosaccharide derivatives in which their particular components are actually being altered, or you can say changed, right? So in this case, number one monosaccharide derivative come is called glycosides. Glycosides. So in this case, what happens? Your sugar molecule, actually it will react with either alcohol or with the another sugar molecule. it will make a resultant product which is called glycosides. So if it is actually reacting with the another sugar molecule, then it will make like this sugar molecule glycoside, and if it is reacting with the alcohol, then it will make sugar alcohol glycosides. For example, if your sugar molecules here are glucose which are attaching to each other, then these are called glucosides. If there are galactose, they are called galactosides. And so on. Right? So this is the number one derivative in which your sugar molecules are either reacting with each other or either reacting with the alcohols. Next alteration which can be done in the monosaccharide is that the sugar acids. Sugar acids. In this case, your sugar molecules functional group of the sugar molecule that actually being changed into another functional group. For example, if we have a CHO functional group here, then it will change into the carboxylic functional group. Then your sugar molecule is being changed here, right? And this type of molecules are called sugar acids. So if there are change into the aldehyde group, right, at the first carbon, then they are called aldonic acid. Aldonic acids, right? And if this change is occurring in the terminal carbon atom of the sugar, then this one is called uronic acid. If both of the functional group and the terminal group are being actually replaced by the carboxylic acid, then they are called alderic acid. Alderic acids. Right? So in the sugar acids, you have three type of acids. Aldonic acid, when only functional group is being replaced by the carboxylic group. Right? Second one, you have uronic acid, when the terminal carbon of the sugar molecules will be replaced by the carboxylic group. Then you have alderic acid, in which case functional group and your terminal carbon, both of the carbon atoms will be replaced by the carboxylic acid group. So these type of acids are called aldonic acids, uronic acid, and the alderic acid. For example, if there is a change occurring in the glucose molecule in the functional group, then this is called gluconic acid. Gluconic acid, right? And if there is a change occurring in the glucose molecule at the terminal, then this one is called glucuronic acid. And if it is occurring on the both functional group and at the terminal carbon, then these are called glyceric acid. Right? So next sugar derivative or monosaccharide derivative is your alditols. Alditols. In this case also, you actually alters the structure of the monosaccharide, in which you change the functional group CHO aldehyde group into the hydroxyl one, alcohol one, and these type of derivatives are called sugar alcohols. And their examples are sorbitol, xylitol. ठीक है? Fourth type of monosaccharide derivatives are your deoxy sugars. Deoxy sugars. Right? In this case, you actually replace the hydroxyl OH groups into the hydrogen atom. Right? and these type of sugars will become deoxy sugars, and their example is beta D 2 deoxy ribose, which is present in the DNA. So that is a monosaccharide derivative, right? Next derivative are called amino sugars. Next derivatives are your amino sugars. In this case, you actually replace the hydroxyl group with your amino functional group, and these type of sugars are called amino sugars. For example, if you are changing in the glucose, then they are called glucosamine. If you are changing the group into the galactose, then they are called galactosamine, right? So both are the sugar derivatives. So they are the different type of the monosaccharide derivatives which are actually changed or altered into some other type of the sugar molecules. So sometimes question can be asked from here also. So you need to remember everything very clearly, right? So in this lecture, we discussed about the basic general properties of the carbohydrates, how they are actually being synthesized in the plants, their functions, and their classification. And in the classification, we discussed about the first category, which are the monosaccharides. And in the monosaccharides, we discussed its property, how they are being synthesized from the simple triose sugars, right? What type of processes carbohydrates involved, and what type of the derivatives they have. So all these processes are very, very important. Question can be asked from here. So I guess for the today it is okay, right? So I will stop my class here and see you in the next class. Take care.